of the step. And as you remember, I was talking just about gram stain. Gram stain is most common technique methods that we are using in the clinical microbiology to distinguish gram-positive and gram-negative bacteria. This is about the structure of the cell wall of the bacteria. We know that gram-positive cells have a thick layer of the peptidoglycan in the wall cell, and that retains the primary stain. The gram stain methods consist of 4 steps, and we are going to use 3 different stains, step by step. And gram-positive cells, since they have a thick layer peptidoglycan structure, this structure is able to retain the primary stain, which we call crystal violet. Okay, and gram-negative cells have a thinner peptidoglycan layer that allow the crystal violet to wash out on addition of ethanol, which we call coloration. I am going to show you them step by step. Gram-negative cells are stained pink or red by the counter stain. We are using other stains. These stains are commonly either safranin or fuchsin. Okay. Now, here you see the step of the gram stain. Okay, as I said, gram stain, the most common stain technique that we are using in the clinical microbiology to distinguish gram-positive and gram-negative bacteria. To identify the microorganism is the first step in the microbiology. Okay, to examine them under microscope, stain them then examine them under microscope to make a first step of the identification. Now, as you see, there are 4 steps. In the first step, you are going to put a crystal violet as a stain, okay, on the sample, and then let them to stain with this stain, okay? And in the next step, step 2, you put a iodine, okay, in order to fix the stain with bacteria to strengthen the level of the stain. And then on the third step, okay, which is known decolorization. Okay, you decolorize with alcohol or acetone. If bacteria is gram-positive, okay, the crystal violet still stay there. Okay. However, if the bacteria is gram-negative, since the peptidoglycan structure of this kind of bacteria is too thin, and then it will wash out via alcohol or acetone. Okay, and on the third step, you are going to put other stain, which we call counter stain. It can be either safranin or fuchsin, okay? And then gram-negative bacteria will stain with a safranin or fuchsin, and the appearance is, as you see here, pink or red, okay? You are going to do these steps, these of the step of the techniques, and then step by step, okay? At the end of the procedure, if they are gram-positive, you will see these colors, okay? Violet, okay, purple, like. If they are gram-negative, in the step through, due to the decolorization, they will wash out the crystal violet and stain with the safranin, if you put, or with a fuchsin. Okay, now, and the more common example, well-known example for gram-positive bacteria, such as Staphylococcus aureus, okay? If you have a Staphylococcus aureus in your samples, and these bacteria will appear purple, okay, blue. If the sample include gram-negative bacteria such as E. coli, Escherichia coli, the crystal violet iodine complex is washed out during the decolorizing step, and the bacteria are counter-stained with safranin, and the bacteria will appear red, pink color. You see here, okay, now gram stain positive right here on the okay, these are okay, gram, right? Gram positive, purple, or how you can say, and gram negative, okay, on the right here, okay, how you see how look like under microscope. Okay. Okay, this was the most common technique that we are using in the clinical microbiology. And then after the first step of identification, you have to cultivate your samples, okay, which we call, okay, bacterial culture, okay, now bacterial cultivation. There are primary culture, subculture, and pure culture here. And when culturing specimen, the primary culture plates are inoculated using the dilation stick techniques. Did I show you before the dilation stick techniques? I guess yes. Okay. To obtain isolated colonies, if the colonies of the primary plates appears as mixture of more than one species of bacteria, a subculture must be performed to separate these different species. And to grow the growth of the several colonies originating from a single colony, which is also known as single cell, is known as pure culture. Okay, if you, after subculture, you separate these different species from each other, and then you put a single cell on a plate, let them to grow, is known pure culture. Only one single cells is grow on in the medium, in the appropriate medium, is known pure culture. Okay, now primary culture, if needed, subculture, and then pure culture is there are different type of bacterial cultivation. Now you see here the pure culture of the Staphylococcus aureus. Each colony began as an individual parent's bacterial cells. Okay, all these are known a colony. Okay, colony of the bacteria. They started with a single cell, single bacteria, and then they will grow and become visible. Now, the appearance of the plate sometimes is help us to differentiate the bacteria. I am going to show you just in a few minutes. Okay, now when it comes to the type of the culture media, okay, we put bacteria, microorganism in a culture media, okay, and many microorganisms may be grown in a laboratory on artificial media, okay, which we call culture media, okay, to grow, okay, or culture microorganism, it is necessary to provide proper nutrients and growth conditions. Okay, sometimes you need to let them to grow faster than normal, okay, to save time. Okay, the growth of the microorganism on artificial material is referred to as culture, or we use plating them, culturing them, cultivating them, and the mixture of the nutrients on which microorganism is grown is referred to culture medium. Okay, the isolation and identification of viable pathogenic organisms on culture media is the standard for diagnosis of many infection agents. Okay, first, you identify, in the first step, you identify under microscope. Sometimes you stain them and then see whether is gram negative, gram positive, and then you choose the appropriate medium, okay, to isolate them and identify them in advance. Now, for that, you are going to use a culture medium which contain appropriate nutrients, okay, a appropriate condition for each specific microorganism to grow as needed. Now, requirements for bacterial cultivation are several, and oxygen requirements. Okay, some bacteria does like oxygen, some doesn't. Okay, and nutrients. Sometimes you need to put a specific nutrients to the certain microorganism. Okay, temperature, as we told before. Most of the bacteria, they like to grow at 37 Celsius degree, but there, a few bacteria, they like grow around 30, okay, some of them about 40 Celsius degree. pH, important. We should also create a sterile condition moisture, okay, humidity of the plate also is important, and carbon dioxide rate also important. These all conditions should be justified based on the microorganism that we expected to be in the sample. Okay. Now, as you see, there are several types of the culture media here. Okay, these can be either solid, okay, or liquid, and the content of these medium may be a little bit different from each other. Okay, and we have to know, okay, which medium you should use for a microorganism that you expected to be in the patient samples. As you see in the media of this type consists of specific amino acids, sugar, salt, vitamin, minerals needed to ensure the proper growth of certain bacteria. Sometimes you have to put an extra nutrients based on the type of the bacteria. Okay, we are fortunate. These all culture media are commercially prepared, are ready for use, and disposable. We have disposable culture plates and tubes, liquid tubes, and you use them and then throw out. Okay, now, as I said, the requirements may be differ from each other, such as oxygen requirements. Okay, based on the oxygen requirements, we classify bacteria as aerobes and anaerobes. Aerobes, which grow with oxygen, okay, anaerobes, which doesn't like oxygen, they grow without oxygens. Most clinically significant aerobes are really facultative anaerobes. What that means, they can grow either aerobic and anaerobic conditions. Okay, the microorganism which can grow in both condition, okay, we call facultative anaerobe microorganism. Okay, you have to know whether they are anaerobe or facultative anaerobe, and then set up the condition, the culture medium, okay, according this information. Now, incubation after you cultivate the bacteria in a plate, on a plate or in a tube, then you have to incubate them, right? In order to let the bacteria grow. Okay, the bacteria grow on artificial media or in a culture typically requires 48 hours. However, for anaerobic cultures, we need more times, maybe 3 days to 5 days. Okay, there are different types of media. Therefore, we classify the media such as enrichment media, contain factors such as carbohydrate that give colonies a particular organism distinctive characteristics. Okay, and then we will be able to differentiate them. Okay, this is based on the scientific information. In some we know that some bacteria are able to use lactose, some does not. Okay, and when they use a lactose, they will give a different color. And then you put a lactose on in the media, and then let them to give us a different color on the plate, then you will be able to differentiate them. Okay. Now, supportive media contain nutrient permitting the growth of the non-fastidious organisms, the organisms that are not able to grow faster. The supportive media help them to grow faster. Okay? And this type of media do not give one organism any growth advantage over another. Okay? It support all of them. Okay. Okay. Excuse me, I have to answer my phone, but it's okay. Okay, now selective media are prepared by adding dyes, antibiotics, or other chemical compounds that inhibit certain bacteria while allowing others to grow. Okay, this is different from the others because, as the name implies, is a selective media. It's allow a certain bacteria to grow while inhibit others. Differential media contain nutrients, energy sources, and color indicators that provide distinctive and easily recognizable characteristics as a result of the microorganism metabolics and biochemical properties. Okay, you have to know the metabolism of the bacteria and biochemical properties, and then put a specific material in the media in order to distinguish them. Okay, as you see, there are several types of the media. Chocolate agar, okay, congee, okay, and sheep blood agar, thyromartin agar. These are maybe most common media, type of media, but we argue this, I mean, all these medias, and there are a few other medias that I didn't put here, are using in the clinical microbiology, and some of them may be specific to a certain type of the microorganisms. We have to know, okay, this information in order to use a proper media for a certain microorganism. Now I'm going to give you a most common example of these medias. As you see here, the McConkey agar, okay, which is a selective and differential medium. And use it frequently for primary plating of specimen to isolate and differentiate gram-negative bacilli. Okay, there are several gram-negative bacteria. Yeah, I mean you have to differentiate gram-positive from gram-negative, but you also have to differentiate the gram-negative bacilli from each other. And as you see here, the McConaughey agar let you to differentiate the gram-negative bacilli, because there are gram-negative bacilli that ferment lactose, such as E. coli, will appear pinky, okay? And there are also gram-negative bacilli that do not ferment lactose, will appear clear, okay, or slightly pink. Here, okay, the slightly pink colonies, and here you see the clear, okay, non-colored, okay, colonies, okay. And then you know that E. coli only able to use lactose, and then you will be able to say that these are E. coli's, okay, growing on these plates. Okay, the hemolysis, the type of hemolysis by bacteria also help us to distinguish bacteria. If you plate them on sheep blood agar, and as you see here, there are 3 different type of hemolysis here. This is alpha hemolysis, beta hemolysis, and gamma hemolysis. And we know that alpha hemolysis is created by Streptococcus pneumonia, okay, which cause of pneumonia disease, okay, a disease of lungs, respiratory disease, okay. And beta hemolysis is created by Staphylococcus aureus, and gamma hemolysis is created by Enterococcus fecalis. Okay, if you are able to differentiate this type of hemolysis, you will be able to diagnose, to identify the agents, whether it's Enterococcus, whether it's Staphylococcus or Streptococcus pneumonia. Okay, now a chocolate agar is an enrichment media, permits bacteria pathogen to grow in presence of a specific essential nutrients. We have other type of enrichment media, such as you see an example of it is buffered charcoal yeast extract agar. This is other type of the enrichment medium that support the growth of the Legionella pneumophilia, the causative agents of Legionnaires disease. We also call Legionella pneumonia, okay? Okay, selective media, as I give an example before, are prepared by adding dyes, antibiotics, or some chemicals, and then allow some certain bacteria to grow while other not. And McConaughey agar is a selective medium, contain crystal violet and bile salt. These inhibit the growth of the gram-positive bacteria. Okay, and then as you see here, okay, this is a gram negative, okay, and this is gram-positive bacteria. I'm going to show you in the next one, maybe here, okay. Yeah, I mean, both of them are gram-negative, okay. However, as you see here, okay, a gram-negative bacilli that ferment lactose, okay, appears pink, as I showed you before, and gram-negative bacilli that do not ferment lactose, okay, slightly pinky or clear. Any questions so far? These are a nice, colorful lecture, as you see. Okay, now it is time to talk about different different type of the samples that we're culturing, such as urine culture, okay, sputum culture, throat culture, because the causative agents for each culture may be different. Okay, if you receive urine culture, okay, you may you know that which kind of microorganisms growing in urines. What are the most common cause of the urinary tract infections? Okay, and that allow you to choose a appropriate culture. Okay, now the culture are done in urine to diagnose bacterial infections. Okay, if this culture is with a in a conjunction with leukocytes, okay, you receive the urine, okay, you culture them in on an appropriate media, and at the same time you examine this urine under microscope to see whether there are some crystals, some blood cells such as erythrocytes, leukocytes. If you examine that there are leukocytes in urines, and then the culture is positive, then this is really meaningful results. Okay, if both of them are together. Okay, okay, urinary tracts infections classify as a lower urinary tract infections and upper urinary tract infections. Okay, if is okay, the infection of the bladder, urethra, such as cystitis. Cystitis is an infection of the bladder, urinary bladder, is known as lower UTI. Upper UTI is infection of ureters, kidney, such as pyelonephritis or pyelonephritis. Nephritis is a infection of the ureters. Okay, the type of the specimen is important. The number of potential pathogen in urine culture is important. You may see a few of pathogens, but it doesn't make sense. For example, for E. coli, to report them as E. coli infection, urinary tract infection caused by E. coli, the number of the colony should be above 100,000. Okay, but for some microorganisms, a few of them, I mean, help you to diagnose as a urinary tract infections. Okay, after you complete a workup of identification of the organism, then you have to also perform a susceptibility test, which we call an antibiogram. Okay, now in some case, in the culture, on the culture medium, they grow more than one microorganisms. 3 or more microorganism types with non-predominating form of the clean catch midstream specimens. The clean catch midstream stream specimens, do you know that? Okay, if you look at a urine sample, okay, you have to also explain the patient how to obtain these specimens. Okay, first, it should be cleansed, the genital area, and then let them, the first urination should be goes into toilet, and then the middle part of the urine is collected into the sterile container for a urine culture. And then this is important to prevent contaminations. Okay, if there are 3 or more organisms on the culture place, okay, which suggests a contamination, which means the patients didn't obtain the urine sample correctly. Okay. Okay, there are different techniques that we detect urinary tract infections. Okay, rapid screening test strip or quantitative culture methods, such as plate method first, and then perform culture, the antibiogram. Okay, the urine culture, the techniques that we plating the urine sample. As you see, the method for inserting a calibrated loop into urine, okay, to these calibrated loops, they ensure that the proper amount of the specimen will adhere to the loop, and then you will be able to culture at the same amount of the urine each times. Okay, now as you see here, that may be helpful to understand how we plating, how we cultivating the urine sample.
The quantitative urine culture is an example of a direct or primary culture where we're setting up a specimen on the plate rather than a colony from a plate onto another plate. The first step is to label your plate, and most often in the lab you'll have a pressure-sensitive label you can just paste on here. The minimum information you need to have on the plate is the last name of the patient, and we're just making up a name here. And then the last 4 digits of the accession number, we're just going to make up a number 2351. Today's date, and the source of the culture. This particular one is a urine. So I'm just going to put UR for urine, and we're going to be using a 0.001 ml calibrated loop. So I put 0.001 on the plate. Then you're going to get a disposable calibrated loop, 0.001 ml, and you want to just shake some out of the container like this and then reseal it to maintain sterility. And then mix your specimen good. Take the lid off. And then you just want to immerse the loop in the urine. You don't want to dip it all the way up here because then urine will run down and you'll have more than the calibrated amount. So just stick it into the urine just like that. Bring it out, put your lid back on, and then hold your loop and your plate just like this. And you're going to make one line of inoculum across the plate. Just like that. Halfway between the divider and the edge of the plate. Then angle your loop this direction, and we're going to make really nice tight streaks all across that inoculum, all the way across the plate. We want to spread the urine out evenly over the entire plate. So we're going to make nice tight streaks across that inoculum. Now about here it's hard to get into the corners, so I'm going to turn my plate around and come back the other way.
Okay. Okay, now as you see, we also are using the same techniques for urine cultures. Okay, however, in the videos, they divided the plates into two different culture medium in order to save money, to save materials. You can do that, but if you use one plate for one culture mediums, then you stick them just in the middle of the place, and then you will then stick them across the first line in order to dilate the microorganism, to let them to grow enough. Okay, the second type of the culture is throat culture. The throat culture is also important to differentiate group A beta and beta hemolytic streptococcal sore throat pharyngitis from a viral throat infections. It is important because the infection, if the infection of the sore throat is untreated, the sequel may occur in some patients, and then they leading to acute glomerulonephritis, scarlet fever, acute rheumatic fever, followed by a chronic rheumatic heart disease, which will affect the wall of the hearts, especially mitral wall will affected by such a kind of the beta hemolytic streptococcal infections. And therefore, it is important to differentiate such kind of bacterial infection from viral throat infections. Here you see the techniques that we obtain throat culture. Okay, you have to use a sterile swabs, and then pass the swab over the rear pharyngeal wall. Okay, here, and also the tonsillar area, and in order to get enough sample for throat cultures. the methods for detection of group A beta hemolytic streptococcus are important because it has some post-infection complications such as scarlet fever or rheumatic disease, and therefore it is important to identify them, to diagnose them. The rapid detections methods, which is a non-culture techniques that we are using. Okay, I am going to give a break after this slides. And we also culturing this kind of streptococcus on sheep blood agars, okay, in order to interpreting the result of the trad culture plays. The appearance of hemolysis is important. Beta hemolytic hemolysis, okay. Identification of a group A beta hemolytic streptococci via sheep blood agar is important. We also are performing some serology tests in order to diagnose such kind of infection, okay, and putting a bile or some kind of other chemicals such as optogenes. These are a kind of susceptibility test, okay, for beta hemolytic streptococci, and also there is a molecular diagnostic testing for streptococci.